And we're live. Everyone can say hi. And we've been going over transcription. And one of the key points, of course, is comparing prokaryotic to eukaryotic transcription. And we're going to finish up with our eukaryotic transcription and some of the differences, and then get into taking RNA and turning that into protein, that is translation, which is a bit more complicated perhaps, but we'll make our way through it. So a couple of key points that we can really point to, or hammer in. The prokaryotic system, in general, is simpler. So prokaryotes, we have alpha 2, beta prime is the core enzyme, as we say, and this is able to carry out elongation and termination, but it can't carry out initiation. In order to have initiation, we have to have the sigma factor. Sigma is what takes the RNA polymerase from being a generic DNA binding protein to being a DNA binding protein, or protein complex, that binds specifically to promoters. So the sigma factor determines promoter specificity. So one of the things I like to think is that as we're going through this, we begin to develop somewhat more nuanced, sophisticated definitions of things. Or maybe simpler. A promoter is simply a site that RNA polymerase can bind to. So if you establish a promoter, RNA polymerase can bind to it, whoop, it begins transcribing. And really, using modern molecular recombinant DNA techniques, any piece of DNA you hand me, I can turn into RNA simply by putting it behind a promoter sequence. And this is case in point, the Moderna and Pfizer vaccines are RNA-based vaccines. And I actually, last year was reading a little bit about the development of these things, and this technology has been around since the early 90s. But you have, and you do this in bacteria, or actually in a cell-free extract if you want to, where you've got the T7, which is a bacteriophage, RNA polymerase binding site, i.e. the T7 promoter, and it's about 24, 25 nucleotides. You have that, and it's actually part of a plasmid. You then clone in whatever piece of DNA you want transcribed. In this case, we're using the gene for the spike protein. And really, in order to do this, you don't even need cells. What you do is you grow this bacteria up, you isolate the plasmid, and it's generally good manners to do a restriction digest where you cut there so you don't make circles. You take this piece of DNA, this DNA template, put in the T7 RNA polymerase, which you can buy, I got some upstairs if you want it, and the nucleotides, a little bit of buffer, and put off to the races you go. And you let this go for an hour, and you can make literally micrograms of RNA, which in the molecular biology world, a microgram is a ton of RNA. And I don't know how many micrograms of RNA is in the vaccine. Actually, it would be an interesting number to know. So you can make RNA completely in vitro. No cell systems at all, nothing to it. And I was reading something a little bit about the issue of how are they removing the DNA template and what they're doing. So there's some downstream processing that they're doing. But I'll tell you that in terms of vaccine production, man, this is like amazing, because it is nothing. It is a couple of days to change this sequence. And in big boy labs, it's a few hours. They can. So if there is a significant variant, you can very easily change the RNA sequence that you're generating to match that. And you make a bunch. The only thing you have floating in this solution is a ton of RNA, some nucleotides, T7 RNA polymerase, which is completely benign, and the actual DNA template, a little bit of it. And so that's easy to purify away, throw in a little bit of DNase if you need to, and boom, you've made RNA. And if we think about other, the sort of more traditional vaccine approach, you take the virus and you passage it through cells a few times to actually mutate it so it's not particularly infectious, and then you inject that. But it's not quite the same as the authentic virus. It works. The other thing you can do is you grow the virus up, and then you autoclave the whole thing to denature it. And then you're injecting somebody with all these denatured proteins, and it works, but perhaps not as good as having a three-dimensional shape of a normal functioning protein. Let's see, I guess it's Johnson & Johnson, or the AstraZeneca. They actually have taken the spike protein, putting it into a non-pathogenic, probably non-replicating virus, an adenovirus, and injecting that. And really all that is a mechanism by which you can get the nucleic acid sequence for the spike into the cell. But compared to this, that's like a really long way around the road. I mean, if you can just get the RNA into the cell and produce this protein, this is, man, the way to go. I mean, it really is a transformative technology in terms of virus production. So that's a cool thing. To make a protein is a lot more complicated, as we're about to see. There is another vaccine that's on the market that is kind of another modern traditional way, where you take the gene, put it into a system like this, but rather than having it just completely floating about with just the RNA polymerase, you've got it in a cell, and you now transcribe the RNA and then translate it. And in vitro translation is a thing that we'll talk about a little bit. It does work, where you can take RNA and put it into a test tube with a lysate of rabbit red blood cells. We'll get into this when we start talking transcription. And it works, but you don't make much protein at all. And you have a ton of other stuff in there. So making protein in vitro, nah. But making RNA in vitro, piece of cake. So upstairs at the moment, we are about to take a different tack, making a piece of the Zika virus. In part, it's just been a molecular biology project that I've been wanting to do. The legitimate reason for it is Will Coxon's got tons of raptor blood, and we can do a very simple assay to see if they are producing antibodies against this Zika virus protein, indicating that they have been exposed to Zika virus, which is an interesting thing. Zika virus is like old news. It doesn't seem to be catching much. But Zika is a virus that's been found in South America and up into Central America, and it's specifically carried by a mosquito that isn't in the mid and northern tier of the U.S. It kind of this mosquito, Fufuis and Anopheles, regardless, it kind of stops around St. Louis. And one of the thoughts is that as climate change begins to change the temperatures, the grounds for this mosquito might actually start coming up a little bit further. I don't think we've seen a case of Zika virus in the U.S. yet that has been homegrown here. But it's an interesting thing. Anyway, so our idea was with migratory birds, maybe on their southern migration, they're getting bitten by a mosquito, contracting the virus, and the birds are pretty much carriers. They're bringing it up here. It's an interesting thing to see. So from a molecular biology perspective, the ability to take RNA or to take a DNA sequence and turn it into protein is a fairly easy thing to do. So we use these very simple bacterial systems because of that. Like T7 is not even a protein complex. It's a single polypeptide. All right, so sigma factor determines promoter specificity. Promoter really then is just a DNA sequence that binds to an RNA polymerase. I mean, that's a pretty good definition of a promoter. And in the prokaryotic system, in order to bind a promoter, all you need is sigma factor. Initiation starts, sigma factor diffuses away, the RNA polymerase makes the RNA. The RNA is going to have some kind of three-dimensional structure happens at the back end that is part of termination, and the RNA polymerase lifts off. And it can just start right back up again. You know, it's one of the things with this business as well, is you're not destroying the template. I mean, one can envision models in which the template is being eaten up as you're making whatever your next product is. But it's not. So the RNA is being produced. We make lots of RNA. Prokaryotes are brilliant, famous for really fast transcription and translation. And as we'll get into this, you know, because they don't have a nucleus, they can coordinate transcription and translation. You and I are much slower, and so, you know, my whole point I've said before, like when you get injected with the RNA, ouch, that hurts. Oh, I think I'm feeling side effects. No. It's going to take you a minimum of 3 hours to be able to take an RNA and turn that into a protein. So we are much slower when it comes to turning genes on and actually generating the protein from them. We take our time. It's a slow walk. Bacteria, 10 minutes, man. They got the protein up and running. All right, so that's because the prokaryotic system is very simple. The eukaryotic system is a good bit more complex. And first off, we've got RNA polymerase 1, Pol 2, and Pol 3, primarily ribosomal RNA, messenger RNA, tRNA. Just as a little plug, though, for work that goes on in the building, Dr. Smith works on something that are known as small nuclear RNAs, and they are often Pol 3 RNAs. And so these are generally snRNAs. There's a lot of RNAs floating around that we don't really know what they do, but regardless. So right off the bat, we got a lot more complexity. Each one of these polymerases is a bunch of different proteins, and they've got intrinsic binding affinity to their specific promoter and transcription factors. So another key point is that the eukaryotic polymerases have no intrinsic, just on their own, DNA binding affinity. Which, if you think about it, that's kind of an odd thing. We're talking about a enzyme that makes its job is to interact with DNA to make RNA, and it doesn't have binding affinity for DNA. What it does is bind to the whole series of transcription factors. So we've got the TFs, and because we're primarily focusing on RNA polymerase 2, there's TF2, and one of the first ones is TF2D. This is one of the first guys to begin setting up shop on a specific promoter. It's got the TATA binding protein associated with it. This is a protein, TBP, that binds to the TATA box. And so we can begin developing this idea in the same way that we looked at the bacterial promoters. We can develop sort of a best fit or consensus promoter sequence. And this is a sequence that we know has the ability to bind these TFs and set up a little nest for RNA polymerase. And it establishes, okay, the first nucleotide of transcription is right there. So we have like there is a minus 35 site and a minus 10 site. Similar, this is a little point, a bacterial promoter just won't work in a eukaryotic cell. I mean, it just doesn't work. And the same is true. A eukaryotic promoter is not going to work in a bacterium at all. So when you want to do recombinant DNA experiments, if you want to express a eukaryotic gene in bacteria, you got to put it in front, or you got to put it behind some kind of prokaryotic promoter. All right, so we've got then the ability to start the transcript in our eukaryotic cell. The next key difference is, and it's so important that I had to have a bold marker, the next key difference then is going to be RNA processing. So as we were making mention, and I find it lighthearted, I always make myself laugh, the book on prokaryotic RNA processing is extremely thin because there is no RNA processing in prokaryotes. In eukaryotes, it's a great big book, series of books. The RNA processing events, the 5' 7-methylguanosine cap, the 3' poly A tail, and as we say, these are template independent, meaning that the DNA doesn't have 300 A's at the end of the gene. It is put on after the transcript has been generated. And the same with the 5' cap. There's not a G at the beginning of the sequence. It is a thing that's added on. All right, so these are critical. The 5' cap, as we'll see, is critical for translation. The 3' poly A tail is primarily there for RNA stability. If you inhibit the ability of the polyadenylation, the RNAs have a real short half-life. So we like our poly A tails. And last and not least, in fact, most importantly, we have exon intron splicing. So this established a book chapter that's called The Interrupted Gene. Eukaryotic genes can have sequences within them that are not found in the mature messenger RNA. So when you compare the DNA sequence in the genome to the RNA sequence that you find in the cytoplasm, very often they are dramatically different, way shorter, and what you got is exon intron splicing. So just to go back through this a bit, often a way of diagramming a eukaryotic gene is like this. We've got the promoter sequence, we have +1, and then we've got a series of blocks and a series of lines, and these represent then the exons. That's typically what you're talking about. And wait, I'm gonna sneeze. Oh God. All right. So the RNA is then processed, and processing happens in the nucleus, and it happens right after transcription termination. So processing doesn't start occurring, at least the exon intron splicing doesn't occur until the RNA has been completely transcribed. And we then have splicing. And so what we can do is splice this guy to make the mature RNA like that. So we end up with the exons and introns. Introns are spliced out, exons are kept. This becomes the mature RNA. It gets polyadenylated. It has the 7-methylguanosine cap attached to it. That is what gets transported out of the nucleus into the cytoplasm. And that's what's going to get translated. A few more things to say, though, about exon-intron splicing, which really makes this thing sing, is not all genes, or you can have the same gene in different cell types or different times of development be spliced differently. And so this is, in fact, a really cool aspect of eukaryotic gene regulation, is known as differential splicing. And there are a lot of things that can happen. The only rule is you can't go from here over to there. So just as an example here, we have exon 1, exon 2, exon 3, and exon 4. All right, you can't take 3 and put it ahead of 2, but you could have a gene or an RNA that looks like this, where you got 2 and 3. Or you could have exon 1 and 4. So you can't change the order. All you can do is change out you can splice out exons in different ways, but you can't bring anybody over. And differential splicing, theoretically, and in rare cases, genomically, you can have the same gene produce 2 completely different RNAs. Generally, it ain't that much of a difference. A classic thing might be that you'll have a stretch of hydrophobic amino acids coated in this spot that would cause the protein to be membrane bound. You can splice it out, and you can have the rest of the protein is now going to be secreted or soluble. So you can change protein localizations. Another standard thing is you might have some kind of kinase site. The ability to act as a kinase might be present, and then sometimes it's not. So different active sites can be taken out. So the exons are part of the mature RNA and can be part of the open reading frame. Not always, but very often. And so what this means is that one RNA or one gene can generate multiple RNAs, and there are lots of different examples. You know, one of the things that sort of a chart to be looked at in molecular genetics is that as you increase in complexity in eukaryotes, so we think about like going from a yeast up to a mammal, the number of introns, average number of introns in each gene is going way up. So yeast are pretty much our simplest eukaryotes. And the average number of introns per gene is like 1 or 2, which means you really don't do much differential splicing. The average number of introns in a complex eukaryote end up being something like 10 or 11 or 12. And there are some genes that have like 50 different exons. And so you can get all kinds of really squirrely, complicated rearrangements and things. This is a little point. I was working on a gene in flies that we cloned because it was related, homologous, to a muscle transcription factor, a transcription factor that is specific in muscle cells. Turns out that exact same gene was also being used in the nervous system, spliced a little bit differently, and it was serving as a nervous system specific transcription factor. So there's all kinds of really cool regulations and games that are played with exon-intron splicing. The other side of this means that when you're cloning or when you're looking at genomic DNA from a prokaryote, you find the promoter, you find the gene. You can easily find the open reading frame and go, okay, this is the protein that that gene is going to be able to generate. In a eukaryote, you start looking and you have an exon that'll have a portion of an open reading frame translated into protein. Then you'll have a whole stretch that in the genome is just coding for nonsense, not doing anything good, and then that's getting spliced out. And so until you look at the RNA in the cell, you can't know exactly how that gene's being spliced out. And that's a whole different kind of technology, but all kinds of really cool things can happen. And so one of the key points behind exon-intron splicing, the reason I think it's worth jumping up and down about, is the following. Dramatic pause for coffee. The yeast genome has around 10,000 genes. What that means is you sequence the genome, you have, a computer program that looks for promoter sequences and looks for open reading frames, that is, sequences that can be translated. And you come up with about 10,000. Okay. When you look at a fly, that number goes up some to between 15,000 or so, 18,000 genes. So back in like 97 or so, the book, on molecular genetics, what we use called Genes, was estimating that the human genome contained around 100,000 genes. Okay, so I figured that grinding up flies, I was safe because the flies are, almost an order of magnitude less complex. Once the Human Genome Project was completed, it becomes apparent that no, there aren't 100,000 genes in the human genome. There's more like about 25,000 genes in the human genome. And all of a sudden I'm thinking, oh my God, I'm grinding up flies and they are really almost as complicated as I am. What am I doing here? It turns out though, because of differential splicing, we can produce well over 1 million different proteins, and the lowly fly, they can splice as well, but they only produce a paltry 100,000 individual proteins. So I'm back to grinding flies up because I'm an order of magnitude more complicated than they are. I think that's sort of my general rule. If I'm not an order of magnitude more complicated than you are, I'm really not going to grind you up. I mean, I might take your blood, and if you're dead, I might grind you up like a chicken. But so the differential splicing allows for a huge elaboration of the potential, the coding potential in a cell or in an organism. That's a big deal. All right, so that's differential splicing. Now, the next step is turning these RNA sequences into proteins. And, I'm just sort of thinking in my head, things that I skipped. There's a whole section in the chapter about exon-intron splicing. I'm not worrying about that. There is these little splice lariats that form. There are whole enzyme systems that are involved in exon-intron splicing. The main thing is there are invariant nucleotides at the end of an exon and the beginning of the next exon. And if you monkey with those, if you change those through some kind of mutation, then the intron won't be spliced out, and that's very often a showstopper for a gene because introns typically have all kinds of stop codons. They're not going to be in frame, which we're going to get into here in just a moment in translation. So exon-intron splice mutations are a great way to mutate a gene. Just on another little point, we're playing around, and I hope it works, looking at the gene that codes for an enzyme that is the final step in vitamin C biosynthesis. And we have the gene, but it's actually got a mutation, or a couple of mutations in it, and I think one of them is an exon-intron splice junction mutation that prevents us from actually generating a functional protein. Because if you did, you could biosynthesize vitamin C, and there's some distinct advantages to being able to do that. Different organisms have the appropriate gene sequences that they actually can produce a functional gene. Pretty much all vertebrates, anyway, I think, have the gene sequence for the vitamin C biosynthesis enzyme. But in organisms that can't biosynthesize vitamin C, it has got mutations in it, and very often they're exon-intron splice junction mutations. All right, so the next step now, we've got RNA. RNA is lovely, but RNA isn't going to do much for us. We now have to carry out the next step in the dogma, and that is RNA into protein, or the amino acid polymers. Just a fancy way of saying proteins. All right, so this, of course, is translation. And it is, in fact, translation. In that, if we sort of take apart these terms, and in this case, the terms are actually pretty good terms. Transcription is what you think of when you're going, like you're taking dictation. You might transcribe somebody's speech. So they're talking, and you write it down. Or you transcribe one document to another document. You're just copying it. You're transcribing right now. You're not really changing the sort of vocabulary of the system. I'm writing, you're writing, we're all writing. Translation is where we're going to go from one language, or in this case, one biopolymer, to a complete different biopolymer. Translation is a completely different, great big deal now, because we're going from the world of the nucleotide polymer to the world of the amino acid polymer. And there's some big, big changes going on here. Just, from a philosophical perspective, RNA made up of the nucleotides, the sugar phosphate backbone and the base, it has the ability to form up and fold into some interesting shapes. But RNA has only got 4 different nucleotides in it, so the complexity is not all that great. It's pretty much all carrying a very strong net negative charge. We're now going to go into the world of the amino acid polymer, and as we've mentioned, we have, so this is a way of thinking about this. We got 4 nucleotides. We got 20 amino acids. So we got a 5 times increase in complexity here, just in terms of the bits and pieces. But additionally, in addition to having 5 times more pieces, the R groups have this dramatic differences of being polar, nonpolar, positively charged, negatively charged, acidic, basic. We're going from a reasonably negatively charged homogeneous molecule into this gigantic, really weird, dramatically different, able to fold up into amazing structures molecule, the protein. It's a big deal. So the evolution of life on the planet very clearly went through a stage that we would think of as the RNA world, in which RNAs were kind of doing stuff. There is the idea of the RNA enzyme or the ribozyme. But it's only when the system moves into this next level of complexity of all these amino acids that we really start to see life take off and do complicated things and get up and dance. So, a couple of things that we have to think about. First off, the idea of transcription comes about, in around the time of World War II. So in the 40s, the notion of RNA is becoming a pretty clear thing, just from a perspective of history. Prior to World War I, or around the time of World War I, Thomas Hunt Morgan, T.H. Morgan at Caltech, was using fruit flies, and because you can actually look at the chromosomes in fly larvae, they're what's called polytonized. They are great big. I mean, you can't see them with the naked eye, but under a dissecting scope, you can begin to see the cells. I mean, they look like gigantic cells. And this is in the salivary glands. And when you squash them and you put them on a scope, you actually can see the chromosomes because they undergo round after round of DNA replication, but they don't separate out in mitosis. They just stack up regardless. Morgan was able to kind of put together this notion of what he calls the chromosomal basis of inheritance. Mendel had come about in the last part of the 19th century with the idea of inheritance and the notion of the gene, and the idea of alleles and things. But nobody would have imagined that it was nucleic acids that were the template, or that were the information. You know, when you start thinking about the different kinds of molecules that you can extract from a cell, proteins do stuff, like enzymes. Nucleic acid is just a wad of sort of a snot ball. I mean, it doesn't really do anything. And so the step from saying that we've got these inherited factors, Morgan's contribution was these inherited factors are chromosomes. If you would ask Morgan, he probably would tell you, yes, the chromosomes are made up of nucleic acid, but it's probably a scaffold for holding on to proteins. And somehow these proteins, we don't know how they're being replicated, but that's what it is. That nucleic acids don't do anything. So it takes a while, but we start getting evidence that DNA actually is carrying information. And we have things like Oswald Avery and the Hershey-Chase experiment, which we'll talk a little bit about later in the lab. But some really cool experiments that begin to demonstrate that it's not proteins that are able to self-replicate or to provide new information. It is, in fact, DNA. So this is a pretty big deal. The next step begins, okay, if DNA is the base information, how is that being translated into protein? And protein translation becomes the sort of the next really big discovery. In Watson and Crick, we're looking at the double helix. That's a really interesting point in terms of structure. And the last sentence in their Nature paper is something about the mechanisms of replication are become obvious from this, or some sort of intriguing notion about it. And that because of base pairing, they can begin to recognize how you have the ability to replicate a chromosome. The next step, though, is taking that chromosome information and turning it into a protein. And that is not any one single guy. It is a bunch of people working for quite a while, even some folks at U of I, developing tools by which they can begin to decode the genetic code. One of the first things that was developed is this tool that is known as, in vitro translation. And in vitro translation is essentially a cell-free extract that you can put an RNA into, and it will generate a protein. They'll translate it. The standard in vitro translation mix is a rabbit reticulocyte lysate, as it's called. I'm not even sure why I bothered to write that up. Don't worry about it. It is taking rabbit red blood cells, and you very gently lyse them by putting them in a real low ionic strength buffer. They pop open, and it's red and bloody, but it has the ability, it has all the components needed to take an RNA and turn it into a protein. There are a couple of tricks we won't bother to tell you about, but what you can do is essentially program it with an RNA, and it comes out. And so generally when you're doing an in vitro translation system, you throw in a radioactively labeled amino acid, and you watch that get incorporated into the protein. So using the reticulocytes and RNA sequences, people begin to figure out how the genetic code is being translated. And a couple of interesting things to think about, and this is stuff that guys at Cold Spring Harbor Labs were gathering over the summer and mulling over, is the requirement for coding. So let's think about this. If you've got 4 nucleotides, and you were reading and translating one nucleotide at a time, that is, one nucleotide corresponded to one amino acid, that would mean that you had the possibility of coding and using 4 amino acids. Well, we know there are 20. All right, so reading the code one nucleotide at a time, not enough information. It won't do it. Okay, what if we then read the nucleotide sequence 2 nucleotides at a time? And the way you calculate the number of two nucleotide combinations given a total of 4 is the 4 to the exponent of the number of nucleotides you're looking at. So if we're going to look at 4 nucleotides, pairwise combinations, and you can do this for yourself, but I can tell you it is 4 to the second. That's 4 times 4. Okay, that's not bad. That's 16 amino acids. That would probably work. 16 amino acids, if that's what you're constrained to. You would probably have life that would exist as bacteria, fairly simple organisms. We may never be able to get complex eukaryotes. So that's moving in the right direction, but that ain't going to do it. So just to keep in mind, this is 1 nucleotide equals 1 amino acid. And now we got here 2 nucleotides equals 1 amino acid. But you don't have sufficient coding potential here. What if we are saying that 3 nucleotides equals 1 amino acid? So now we've got 4 to the third combinations. This is 64 combinations. And hey, that's going to work. So if we say 3 nucleotides equals 1 amino acid, now you got sufficient coding capacity, sufficient information stream to make that work. And so what's really pretty cool about this is you got 64 3 nucleotide combination possibilities. You only got 20 amino acids. We're fat. We're good. We got enough that we can code for every single amino acid. Plus, we have redundancy, which we'll see is kind of nice from a mutation standpoint. You can pick up mutations and the nucleotides and quite likely not change the amino acid sequence. That's good. It also gives us the ability to have stop codons, 64 nucleotides or 64 combinations. We're fat. We're set. We can now begin translating. And that's what we shall do on Monday. Join me, won't you? That's all I got to say. All right.